Okay, this is the Professor, CBMD PhD, and today is Wednesday, July 26, 2023. It is 6:02 and 41 seconds, AM Eastern Standard Time, and today's research, embryonic development. So let's come to it here. The key mechanism in embryonic development revealed, pivotal role of DNA copy number, date July 19th, 2023. Source, Institute for Research in Biomedicine, Barcelona. Researchers have revealed the relationship between autophagy and polyploidy, the latter a phenomenon in which cells contain multiple copies of genetic material. In this regard, they have discovered a scenario where the level of autophagy is much higher in cells with several copies of DNA, and that it can even trigger this programmed cell death. During development and associated processes, the programmed death of certain cells plays an essential role in shaping organs and allowing proper growth. As this cell death is in some cases an effective autophagy, a key cellular process responsible for the degradation and recycling of damaged or unwanted cellular components, and which, taken to the extreme, may end up eliminating the cell itself. As polyploidy is a common phenomenon in many species, including humans, and understanding its implications for embryonic development could have a significant impact on medicine. The progenitor cells can give rise to different cell types. In this case, the Drosophila melanogaster fly, the animal model used in this study, the progenitor cells are larval cells that conserve the capacity to give rise to the adult organism. As these cells do not present polyploidy, they have a single copy of the set of chromosomes, and this allows them to survive metamorphosis and form part of the adult. As in the case of cancer, it is common for cells to have several copies of DNA polyploidy, and this property might confer increased resistance, for example, to chemotherapy treatment, and this is why it is so important to understand this process. And to study this process, the researchers work with the trachea of the Drosophila melanogaster fly as a model which presents a variety of cells of the same type, some with polyploidy and others without. Working with the trachea of the Drosophila melanogaster fly has allowed them to compare cells of the same type, some presenting polyploidy and others not, and to see how polyploidy correlates with cell death during metamorphosis. The research team is now working to determine whether polyploidy cells respond better to stress situations, and, if so, what the mechanisms underlie this response are. And this is the Professor, Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoy listening. Until next time, have an awesome day.